Good afternoon, everyone. Thanks for joining the workshop today. My name is Jia-Ren Lin. I'm from Harvard Medical School. Today, I want to share you our experience with using multiplex image to probe disease or normal tissue. So first, I want to just conceptually asking why we need to using a multiplex single cell image or tissue image. So you can ask why you need to use that. And also the second question will be how you achieve multiplex tissue image or single cell image. And then will be what's the outcome of your single cell image result. So today I want to, hopefully we can cover all of this within 50 minutes, and then we can start to introduce you more what we have done in the past few years using multiplex image. So as the outline like this here. So maybe the easiest thing we can think about why we need to use the multiplex, that's mean multiple biomarker at the same time, is mainly because the heterogeneity in normal and the diseased tissue. And then of course, there are multiple ways to do it. So today I will mainly focus on our developer of cyclic immunofluorescence, or we call it CyCit, and some of the software we've been developing here we're using for image processing and visualization. And finally, what can we get from the multiplex image? Although I will still focus on the cyclic immunofluorescence, but not limited to that. So I will briefly introduce what's the available high-dimensional single-cell analysis tool, and also some user case can show what's the biology you can get if you're not just doing one at a time. So first, just a brief introduction, and I'm using the slide right here from the Human Tumor Atlas Network that's got found by NCI. So the goal of building a human Atlas network is mainly driven by this picture show here is microenvironment of tumor or even the normal tissue you can envision here. It's quite complicated. You have multiple cell types that's occupied in a specific order that interact with each other. And while the traditional method it's either using the single cell sequence or the single color image that's try to pinpoint this complex behavior. But past 10 years, we start to have the power to do a multiple marker at the same time. So that will allow us the first to identify the cell type within a given environment. And second is try to understand the difference between the disease and especially the progression of disease, as well as the individual difference in different patients. So I just try to show a very simple example here, and this is a demonstration. Again, if you can envision there are a tumor cell within a given patient, even within a single genetic background or multiple genetic background, you can start to see heterogeneity on a tissue level that here just demonstrated by the traditional histology stain. But if you go into detail, you can envision there are different clone of the cell or different epigenetic composition. And while you just look at the three different single color or three different fluorescence right here, one is the nucleus stain that's labeled with blue, and second is a phosphorylation of S6, that's a downstream marker or the NTO pathway, as well as another marker for EBP1 that's also downstream of NTO. You start to see the first is the huge heterogeneity between sample to sample, as well as the difference between, although supposed to be the same downstream target of NTO, but they show quite a difference right here. So, and this will become even more complicated if you have power to do more things. Here is one of the data we show right here is a human tonsil tissue. And right here, you under my cursor, you see here is a germinal center that's full of some cell type that's supposed to be PD1 positive and the PD-L1 positive. And these two happen to be a very important player in the immune checkpoint. And as you can see here, they kind of special organized in a very defined way, as well as other cell type, for example, CD8 positive cell that's outside the germinal center, as well as CD68 positive cell that's supposed to be expressed PD-L1 within this germinal center organization. So that's just a simple example to demonstrate what you can see if you're not just limited by your tool of single color or like two color image. So now you have ability to probe multiple or complex cell-to-cell behavior. So first question we can ask, of course, is what kind of composition within this microenvironment? And the second is what kind of distribution of these things that's basically encoded by the spatial information. And third is what kind of the interaction between the independent cells. So I hope this introduction can lay out like our topic today, like that you understand what's the importance for using complex image methodology like SciSIF we use here. And not emphasize even more is if we not only the academia is start to develop more and more methods to do this, a lot of companies start to thinking about this will be an important future to study cancer and normal biology. So there are a lot of investment here, either on the instrument, on the analysis tool, as well as in reagent. So all this just a part of the big community here we can use for multiplex image analysis, especially focus on the tissue or pathology or histology. So I just, this is kind of my thinking of what's when you try to do a tissue multiplex image, what kind of the criteria you try to have in mind, right? So there are several points you need to consider if you want to do a high multiplex tissue image. So it's so first thing is how many marker you aim for. So that depends on the technology itself. And also what kind of sensitivity wise you can accept, because you can envision if you want to detect a lot of markers simultaneously, you might be sacrificed in terms of sensitivity, and also resolution wise, you might need to try to come up with a balance. And the last one other thing you need to consider is the throughput, because a lot of methods, although can achieve very high number of the marker, but they will go very, very slow. So the throughput wise is one other thing you need to consider. And then not to say a lot of these methods quite expensive. So you might need to think about how much we've been communicate with all this company, and sometimes you can cost several $1,000s per slide. So that's why, and at least some of the contemporary method that has their strengths and the weakness in terms of all these criteria. But a more complicated list is right here. That's hopefully it's not most up to date, but cover most of the method we've been able to use nowadays for the highly multiplex tissue image. And again, like I say, we probably today will focus on one of the methods we developed in-house right here, called cyclic immunofluorescence or SciSIF. But we've been able to categorize this image method into several different categories, although all of them probably all use the antibody-based method, but they will be able to resolve this antibody with different way. Like, for example, using SciSIF, you do a dye removal, a repeated image. Some of them will use the DNA barcode to do the multiplex, and some of them will use, for example, like metal barcode, RNC, and maybe. Okay, so without further ado, I just want to briefly introduce the method we developed in-house called cyclic immunofluorescence. And here is just a short video to show you the principle and what kind of thing you can do with cyclic immunofluorescence. Yeah. Sorry. Hopefully, this is a very brief introduction to show you how the cyclic immunofluorescence work. But that we actually developed this cyclic immunofluorescence method earlier before we do the tissue image, where we using the cell culture in a tissue in a plate to do a cyclic immunofluorescence. That's our first paper published a few years ago. And the good thing is they using the same principle almost by repeated staining and remove fluorescence. Now you can achieve high dimensional image, as you can see the illustration right here. So just again, just quickly using a movie to show you, if in a cultural plate, you can start to using multiple marker. In here is focused on cell cycle regulation. Now you can start not only just looking at the one or few particular cell cycle regulator, but the overall cell cycle distribution in a given population. So now you can start to ask a question about how the regulation or how drug perturbation will have effect on this cell cycle program. And we also use the same principle on the liquid biopsy. In here is the blood sample that we collaborate with our outside collaborator real side. They have a method to collect the Buffy or the immune cell in the blood. Then we can spread on the slide. Then we'll go through the same zip workflow. Then you can get high dimensional information from that. And here just a few examples to show you. Now you can use this almost like an image cytometry to study. And also, even more than that, we can start with using some other marker that rely on the mucus or translocation of mucus and the cytoplasm. In here is an example of the NF-κB. So now, unlike the normal flow cytometry, you don't have the resolution to do that. In our workflow, you can actually distinguish this one. And not only that, we've been able to combine the CySIF with RNA fish in this particular liquid biopsy. So now you can use the RNA target plus the protein target to do the same study. And again, just come back to our topic today. We now develop the method to do a tissue-based size image that can using the FFP sample and doing high-quality image on the single-cell resolution. And again, like I say, it's a similar principle we are using. So basically, you do a traditional direct immunofluorescence, then you can do fluorescence removal using the very simple solution. Here is the hydrogen peroxide solution. Then you can repeat this multiple cycle. So here, just a quick video, just show you how the experimental workflow. And you can see we use the automatic scanner and the other instrument to help to achieve this highly multiplex image. Okay, as you can see here, all the procedure is quite straightforward and simple. I think that's another advantage to using the sizes is nothing proprietary. Although we do use some like automatic image and tissue processor to increase our throughput. But if you want to use a more semi-automatic or manual way to do that, that's totally possible. So by using this method, we can achieve the whole slide image. As you can see here, the scale bar right here, we can image multiple tissue in full scale, as well as run into a single cell image to see how the single cell expression of different marker. So even further, we can, because this is nothing different from the traditional immunofluorescence. So you can put on a confocal microscope. Right here is just show you a mitotic spindle that's labeled by the Aurora kinase phosphorylation, as well as some other nucleus structure, for example, LAMI. And we can go even one step further. This is from ONX, so super resolution image. Now you can see even more detail from all this nucleus structure protein. So I probably will quick go over this. Yeah, so with this and with some very careful experiment thing. Sorry. Things has some problem, I apologize, but I think we can still go over to the same slide. We can achieve very highly pressed image in a reasonable amount of time, and like I suggested earlier. So again, so we can still get very nice result, and as well as we can combine multiple different modality. In here, just show you using RNA-fish and Decisive together. Okay, so but most important, I want to emphasize a little bit is how we use to validate our antibody. We did not actually try to make this antibody ourselves. All the antibody we are using in the experiment is purchased from commercial vendor. So we need to go through a very rigorous antibody validation step. And then in here, just a few criteria that we use for validation. Since a lot of antibody we're using here is direct on the human tissue, so we don't have the luxury to do some knockout or some control experiment like we do in the cell line. Fortunately, a lot of them been validated with vendor by either cell line or other form. But we also need to do, but all this vendor is using testing antibody in a very given condition and a very specific, for example, tissue or cell type. So if we want to do that in a different tissue cell type, because it's not only the on-target activity we need to consider, most likely we need to consider the off-target activity. So we're using several criteria. For example, we look at the most large-scale tissue distribution and localization to see if this is what the expected pattern. And now we can really one step further to see what kind of cell specificity and subcellular localization we see. And most importantly, since we have the ability to label multiple color in the same sample, now we can start to see based on our prior knowledge, what kind of marker co-correlation or co-segregation of marker we should expect to see. So we can using the marker that we have higher confidence to validate the marker we have less familiar with. So the other things we try to do recently is we were doing a cross validation using the serial section. So we do a Decisive on one section, and using the serial section to do a traditional RC. That make us more confident our result will reflect what traditionally being used as well as the clinical being used antibody or performers. And then we can apply this to immune profiling or understand the immune system in tumor or disease or even normal situation. So just try to do a quick sum up of why we choose to use Decisive. I think it's, like I say, Decisive is compatible with multiple different formats. We can do a high content or cultural cell, and we can do liquid biopsy as well as FFP tissue. So the same workflow, even we can move from more preclinical research to the clinical research. That's make us a very unique or very convenient format. Not to say this is probably few open source or the non-commercial platform you can use for multiplex image. Okay, so just quickly review a history thing. So we started probably 6 or 7 years ago when we start with the cultural plate. Now we shift to tissue image, and we have several milestones as well as some of them I will probably share with you. We now is part of the H10 consortium and funded by multiple institutes. So that allows us to collaborate with more outside or inside collaborator, and we can start to learn more about this tissue heterogeneity using Decisive. Okay, so alongside with the experiment, we actually developed multiple software to help us to either process this image or share this image. So here I will focus on two of them. One is we using for the tissue image processing. And the main question is, since most our research here is require we do a whole slide image scan, as you can see right here. So and the image will acquire tire by tire, like I show here, but also we acquire from different cycle. So try to assemble this into a single image that allow us to do the visualization and analysis. It's not a trivial question. And fortunately, a very talented software engineer or research in our lab think about this very hard and try to develop a tool called Ashla. So the challenge here is because it's not only you try to stitch multiple tire like within the cycle, but also you try to assemble the tile or the stitch image across different cycle that's into a single image. And if you do that independently, you will end up with non-so optimal result. And our approach here is try to treat this as a one-step method to try to using the first image as the layout and try to lay multiple things down. So that improve very effectively to assemble this image into a single nice analyzable or visualizable image. But with that image, so each of our images probably over several hundred gigabytes in general. So in order to share this image, it's also not so easy, or even that people have ability to open this image. So we want to make this sharing or visualization much easier, as well as we want people can use this image share tool to understand complex data. And also in the end, need to be able to do it on a personal computer, but not supercomputer. So what our solution is, we call the MANOVA or MANOVA story or MANOVA author. And here I will just using a short video to show you how the MANOVA works. Excuse me, I think. Apologize for that. Let me try to see if I can fix the thing. Apologize for interruption. So yeah, like I say, I want to share you the short video about the MANOVA software that we developed here. Thank you. So yeah, thank you for watching this. So as you can see in the video, the vanilla story is a very powerful tool to share your image data as well as the integration of your numerical data. And it's actually built by two independent components. One is the vanilla story, that's the one you just showed, but we also have another tool that allow you to build a vanilla story that's to display. So with this two tools, you can start to build your own vanilla story. And all this is open software, so you can start to download. And if you have any question, here is some reference information as well as the GitHub page. You can start to download this. So now I want to go into use a final part of my talk here to talking about what kind of the analysis you can done with multiplex image data. So although we all know it's not that easy to collect the multiplex image platform, and that there are some limitation of different modality. So there are several different main technology we are using, maybe people are using right now. They all have their different strengths and the weakness. And one very essential is component, as we discussed very beginning, is how many biomarker they can image at the same time, but also the how many number of sample they will process as a given day. And some of them even they cannot image a too large of area like we usually do with SciSafe. So they usually can use tissue microarray. I will introduce that later in my talk as an alternative to achieve more number of samples. And here again, I just want to use a summary table to show some like important paper that's using multiplex image as a main modality to ask some biological question, so we can learn some interest what kind of analysis method they've been used, especially the special encoded analysis method. And the few category we are thinking you can use this kind of spatial encoded information is as easy as like a regional cell count. And one example I will show later is we call it immunoscore, as well as like cell-to-cell interaction or neighborhood analysis. And also something even more complicated, you try to see if there are some special correlation by single marker or multiple marker. And lastly, we want to see some sort of like special organization, also like a community of the cell that's within your given sample. And I won't touch too much today is some even more complicated analysis using fractal or the entropy to try to ask if you have special characteristic in any given sample. And it's, like I summarized earlier, some different groups might use different methods as well as their analysis algorithm. So I just want to pick a few examples to do a quick overview of the spatial analysis. Like I alluded earlier, the immunoscore is very simple idea. Try to look at two different regions. One is the center of tumor, and one is on the margin of tumor. And using two different immune marker, CD3 and CD8. And just by looking at this quite easy apply criteria, although it's not as quantitative because mainly this is done by pathologists, or currently they try to develop a company spin out from the original research group, try to develop a computational approach to do that. And now you can classify your tumor into two different or several different categories, either immune score high or immune score low. And now you can start to see if they will correlate. And in their research, they do show this immunoscore already related to the prognosis. So even with a very simple regional cell count, you already achieve much better than not contain any special information. And the other or more complicated spatial analysis is try to using the cell-to-cell neighborhood analysis, and by define each cell into an independent cell type. Now you start to ask if you see a specific cell-to-cell interaction in this particular sample. If yes, then you start to ask this specific interaction will that correlate with any clinical outcome. And more complicated because if we just do a cell-to-cell analysis, then you kind of ignore the whole community contribution, right? And also it's become a more like one-to-one question. So later people develop a more complicated method by looking at the spatial composition with a given region. And I call it spatial motif analysis and more city block like concept. So in any given tissue sample, you can start to divide into different blocks. And now you see what kind of the spatial pattern within a certain size of block. Then you can start to ask if a merge pattern show up in a specific or a disease or normal tissue. So in our lab, we actually not using that type of analysis. The reason I will define later. A PhD student in our lab, Xu Wang, actually more interested in this question and try to ask more broadly, especially in the whole slide section, what kind of the special correlation of any given marker in a 4-slide scale. And by looking at the distance between a different marker, then you can start to ask, is there at any given difference, how much correlation between you and your surrounding environment in a particular marker. And for example, like here we show a CD1 spatial correlation in different distance, and we now start to see, although within a 10 micron is probably you and your neighbor or you and yourself, it's highly correlated, and this correlation start to drop within a given distance. But even within 30 micrometer, you start to still got 0.5 correlation, Pearson correlation. That's indicate you likely to find a CD31 positive cell within like 30 micron distance. So they indicate some sort of clustering method. And indeed, like looking at the plot, this is a full-scale image. You start to see some pattern, and the yellow indicates a CD1 positive cell. Now you start to see this kind of pattern emerge. And this not only true for one marker, but we also test the multiple marker. So the conclusion, easy conclusion is most biomarker is spatially correlate, no matter what kind of things we test. And this spatial correlation drop over distance, but it depends on which marker you add. They are different in different distance. So that raises an interesting question I want to allude in the last part of my talk is why we need to do a whole cell or whole slide analysis. The reason is actually quite simple, is because if you are thinking you do a special sampling and you for any, it's not actually limited to the question we are asking, but also to some other field of research, for example, like geographic, economy, or even politics. If you, in most of case, you are thinking about either the stochastic or symmetric distribution, and no matter how you sampling, you probably will get a pretty good result from your sample, as you can indicate here is the green and the red box. But if you start to have some sort of pattern or systematic randomness right here, and it depends on where you sample, you start to get a different result. And that's exactly what we see from our research. Like I showed earlier, we do see a very strong spatial correlation in different sample. And in the histology, like sometimes we are limited, although for most of the time we can get a whole slide section or whole tissue image. But for a lot of time, given the limitation of our power to scan or process this sample, we can use something like whole needle biopsy, that's punch biopsy, as many of you might know, or even from the whole slide section, we can reconstruct a tissue microarray by punching a small piece of tissue and rearray into a block, bigger block. So now you can profile multiple samples at the same time. But the challenge here is because you do this kind of special sampling, so you will encounter the question we start to ask earlier. So you choose or the even more simpler is you start to thinking, you start for any kind of tissue image, you start to choose field of view. You will see very different results from very different of view, as I demonstrate here by looking at the green bars and red bars. You see totally different film. Okay, so we are working with NCI sponsor human lattice network, and one of our project focus here is actually asking the question, try to compare different image modality as well as some sort of important criteria within this special image, and one of them happen to be what kind of impact of special sampling on this result. And this is a multi-institute collaboration, and we are try to distribute a colorectal cancer tissue sample in a serial section and send to different center, and each center will use a different image modality to probe. And for us, we actually pick a serial section of this sample and try to using our SciSafe to do a profiling. And what we end up finding is we can actually reconstruct the tissue microarray using the whole slide section virtually, right? So by select the region of interest as I demonstrate here. And even within one sample, we can get a huge amount of difference, as I show 9 different virtual TNA actually only generate from one sample. So that raises our concern. And here is another example to show, just by looking at even more simple than that, we just looking at the different field of view, you start to see a very different picture from one given sample. So although this kind of, we get a very striking result, but we start to asking ourselves if this is real true or it is just due to our virtual version of this process. So we actually get a real tissue microarray and try to image them with the same modality. And the result try to show here, not surprisingly, we also show a very different result from real TNA. And we, one step further, we start to try to look into more detail. This is just 4 different single cell or 4 different tissue microarray from the same patient. In some case, you see quite nice, but you can see right here for patient 45, this is apparent outlier that's not sampled to too much tissue. And here is even worse. That's the heterogeneity within the patient is non-negligible. And if we just looking at the one single marker, and in the end, we try to even address this question of experimentally by go back to the original tissue block and get the whole slide section from this using to construct this TNA, and asking if we do a whole slide image versus the one we using TNA, how much difference can we get? As you can see here, for this is a even best scenario, case scenario of try to evaluate CD8 plus T cell, we start to get a huge variation, although the mean difference is still kind of pretty close to the whole slide section. But in some case, we can get a very wide distribution like we do show up here. So for example, CD31 positive cell in the TNA, we can get a huge distribution right here from even lower from 0.05, so that's 5%, as high as 40%. And if you just do in a random sample from the data, we actually get pretty good correlation. So that's go back to the same idea, like, and I won't go into too much detail. It's actually just a very simple or very old statistical question that people ask. How does special sample, if your sampling is not really random, what kind of consequence will you be? And in the end, it's just this effective sample size is not as you expect it. So even your TNA is sample more than 1,000 cells, your effective sample size probably as low as like 12 cells. So you probably don't have enough statistical power to do that. So I will skip this part given a time constraint, but I will just use the last slide to show a few things. First is this is all the community effort. In here, we have a huge group between ourselves and the collaborator to do the size or try to develop a different method or using SciSafe to asking different questions. And again, some of the tools, some of the concept we provide is not limited to SciSafe, but for the general multiplex image. And here I list some few key reference, but you can find all this in our website, SciSafe.org. So you can, hopefully you can learn or you can find something you think is useful. And again, try to list the people contribute to the project, and especially the one I mentioned in the last, the H10 Sadhana TMP project, and the funding source right here. Although this is the key contributor for that project, like I say, this is a community effort. So this is our SciSafe team and collaborator. So it grew over the past few years. So it's a very strong team and community, and glad I be part of it. So again, just try to list a few of our main funding source right here as the disclosure. So happy to take any question. Thanks for your time.